Hello everyone, ¿Cómo ça va? Hope you're doing well. Today we'll be talking about gel electrophoresis. What is gel electrophoresis, you might ask? Well, it's a biotechnology that can be used to separate out molecules like DNA that you can see on your left and proteins on your right, based on their size or how big they are. Gel electrophoresis involves an electrical field to separate the molecules through a gel, gelatin-like material, or like the jelly that you have for dessert. Now, let's talk about how it works. For a start, you need to have the gel like I've mentioned earlier. It can be either agarose or polyacrylamide gel. The gel allows the DNA molecules to travel within it. Just imagine the DNA is like a guppy fish swimming through the jelly swimming pool. One end of the gel has these holes called wells. You can see them on your left in the red box. The wells are where the DNA is placed or pipetted into. In order to create an electrical field, we have to have a cathode and an anode. The area of the gel where the wells are is negatively charged or the cathode, while the opposite of it is positively charged. Can you guess where the DNA will travel towards? You are absolutely right. The DNA will move towards the positive charge since it is negatively charged. Size of the DNA fragment affects how far it travels. In this video, you can see that small size sample travels further while large sample travels slower and lag behind. There are few more things that you need for you to perform gel electrophoresis. To power the process, you will need power supply that is connected to the electrophoresis tank. In order for the negative charge to flow towards positive charge, you need something that will conduct electricity, which is the buffer, such as TAE buffer. This buffer will completely cover the entire gel. Next step is you will load your DNA sample by pipetting into the wells. And make sure that you have mixed your samples with DNA loading dye so that you can see it's running and track the movement of the bands later on. Finally, you will always load something known as the DNA ladder. This is something that you buy from a company and it tells you the sizes of their fragments so that you can match them with your unknown samples. Finally, to visualize the DNA, the gel will be placed under the UV light and this will show up the stained DNA as white bands. From this diagram, what you can see is that there is a visible band of the sample whereby its size is 400 base pairs. DNA fingerprinting uses gel electrophoresis to distinguish between samples of the genetic materials. This is mostly used in CSI, crime scene investigation, where DNA found at crime scenes is compared to DNA from suspects. Now, I want you all to imagine a murder scene in CSI Miami where Mr. X was brutally murdered in his home by one of these three suspects, Mr. Bland, Mrs. Rose, and Mr. Muse. Unfortunately, the suspect left bloodstains at the scene. So, what the police can do is that they can grab samples of DNA from each of the suspects and the blood from the scene itself. Then they could do DNA fingerprinting on them. Who is the murderer? Who do you think like they are capable of killing someone? The crime lab managed to separate their DNA using gel electrophoresis, and using the gel, we can see that Mr. Bland is found guilty of killing Mr. X. In other words, his blood matches up with the blood found in the crime scene. Oh, poor Mr. X. DNA fingerprinting is also used to establish paternity. This technique makes it possible to determine a child's biological father. We hope this video is helpful for you. Thank you for watching. Bye!